Hello and welcome. My name is Richard Turner and I'm a clinical lecturer in clinical pharmacology from the University of Liverpool in the United Kingdom. And I've been invited to give a talk today entitled Pharmacogenomic Testing in Clinical Practice. I would also just like to give my thanks to the conference organisers for inviting me to give this talk. So by way of an overview for the following talk, I'm going to be covering the following areas. Firstly, the background to the problem, and secondary, considerations for implementing pharmacogenomic into the clinic. Thirdly, just going to do a brief case study of Warfarin as an exemplar for the implementation of pharmacogenomics. And fourthly, going to briefly discuss the barriers to implementation and the ongoing European Ubiquitous Pharmacogenomics initiative. And lastly, just with an eye to the future, I'm going to give an overview of rare variant pharmacogenomics and drug-drug gene interactions. It is clear in clinical practice that there is notable variation between patients and how they respond to a given drug, affecting the drug's efficacy and/or its toxicity. This interindividual variation in drug response is important when you consider the wider context that global spending on medicines is expected to reach $1.5 trillion by 2023, yet it was previously estimated that across a broad range of therapeutic areas, only half to up to three-quarters of patients respond beneficially to first-line pharmacotherapy. Furthermore, 6 to 7% of all hospitalizations are attributable to adverse drug reactions, and 15% of inpatients experience an adverse drug reaction. Therefore, interindividual variation in drug response is important, and it is thus paramount that we understand the factors underlying this variation and seek to put in place mechanisms in order to improve the precision of our prescribing. Drug response in many instances can be considered a complex emergent phenotype, which is multifactorial in origin. Thus, this slide depicts several of the factors which can affect drug response, for example, demographics such as patient's age and sex, or comorbidities such as existing hepatic or renal impairment, environmental factors such as smoking, as well as drug-drug interactions, drug-food interactions, and what we're going to focus on today is genomic variation. But clearly there will be variation in the other omics levels, which will also be important for modulating drug response. So that brings me on to pharmacogenomics, which I define as the study of the role of genomic variation in drug response, and importantly its application into clinical practice. And this has been a very fertile area of research, and now over 200 FDA-approved drugs have genetic information somewhere within their product labeling. And importantly, therapeutic recommendations involving germline genotypes have been developed by international guideline committees now for over 75 drugs. Those two important committees in pharmacogenomics are both CPIC, the Clinical Pharmacogenetics Implementation Consortium, as well as the Dutch Pharmacogenetics Working Group. And this slide also just shows you some notable examples of drug-gene pairs for which there is already a clinical guideline available. Importantly, many genomic variants associated with drug response are relatively common. Thus, when you survey the collective frequency across a panel of actionable pharmacogenes, virtually everybody in the population carries at least one actionable diplotype. Clearly, not everyone in the population will also then be prescribed the drug relevant to their diplotype, but we still expect that about 5% of all prescriptions could potentially be modified by existing pharmacogenomic knowledge. So what are the considerations for introducing pharmacogenomic testing into the clinic? Well, clearly, firstly is which genome are you going to be targeting? So here in terms of pharmacogenomics, we generally focus on the germline genome, but clearly the somatic genome is important in oncology, and potentially the microbiome will become increasingly important. Secondly, what drug-gene associations will you be focusing on, and then within those genes, which variants will you target, and what's the strength of the evidence, and are there existing guidelines already in place for your chosen drug-gene associations? Then in terms of the patient pathway, who should you test and when should you test, and is the testing infrastructure available and how does it work? What's the test turnaround time? What does the report look like? Also, what should you do if you find incidental drug-gene associations, for example, which are relevant to drugs which the patient is already taking? How do you handle those situations? And then in terms of the recommendations, are the alternatives available in your healthcare setting, or are they already off-licensed? You have to, I suppose, ensure that when you implement pharmacogenomics that you understand what's already in place. And so clearly there's already phenotyping strategies for certain enzymes, so for example TPMT. Clearly the knowledge and upskilling of healthcare professions is important for the implementation of any novel technology, and in our era of limited healthcare resources, cost effectiveness is paramount, and the ability to demonstrate cost effectiveness is paramount, I suppose, in order to try and get the green light, and also to demonstrate the utility of pharmacogenomic testing. So what are the indications for pharmacogenomic testing? Well, for the vast majority, pharmacogenomic testing is used as a predictive marker in order to guide dose and drug selection. However, there are some instances where a pharmacogenomic test may be useful in diagnosis, for example, in rare instances of drug-induced liver injury, and also sometimes the recommendations from a pharmacogenomic test may be increased monitoring and surveillance of a patient. Clearly, it is important to consider patient consent for pharmacogenomic testing, and as for any medical consent, it should be informed, voluntary, and the patient should have capacity to make the decision. In the context of pharmacogenomics, it is there to guide treatment once the decision to treat has been made. Pharmacogenomics is not used in order to predict susceptibility to a disease or future disease, and it's not there to diagnose a disease. I see it thus more analogous to renal or liver function testing, which is used every day in order to help us guide dose and drug decision making, and therefore we have to be mindful to avoid genetic exceptionalism. Importantly, the majority of pharmacogenomic genes are also not associated with any diseases. I've highlighted a couple of possible exceptions, such as benign hyperbilirubinemia, Gilbert's syndrome, and potentially the non-synonymous variant in factor V, which gives rise to factor V Leiden, are exceptions to that, and we may need to think a little bit more about those when we're taking consent. But on the whole, pharmacogenomic genes are not associated with underlying diseases. I suppose it is important also to think about, in terms of consent, that pharmacogenomic treatment recommendations will evolve, and thus recommendations for a patient may change as we understand more about their underlying genes and variants. And actually, it is possible, as we understand more about variants of uncertain significance, that a patient's predicted phenotype for a gene may also change, and thus patients should be mindful of that. Clearly, if you are carrying out genomic tests for another indication, then for example, rare disease or cancer diagnosis, then additional consent for those purposes will be required. In order to have a structure in which to better conceptualize pharmacogenomics, personally I find it useful to revert back to some basic pharmacological principles, and in particular focus on pharmacokinetics and pharmacodynamics. So pharmacokinetics is essentially what the body does to a drug, and can be further subdivided into absorption, distribution, metabolism, and elimination. And many notable pharmacogenomic associations are due to genomic variation within either drug metabolizing enzymes, such as cytochrome P450 enzymes, or drug transporters. Secondly, pharmacodynamics is can be clearly defined as what the drug does to the body, and you have both the so-called on-target desired therapeutic response, but also many drugs have undesirable off-target binding, and these can lead to adverse events, and these can be further stratified into non-immune or immune-mediated mechanisms. as provided in the slide. And now I've just gone on to provide some examples of notable pharmacogenomic associations, dividing them into those due to pharmacokinetics, on-target and off-target pharmacodynamic mechanisms. And for the purposes of time, I'm only going to cover the Warfarin case study as an exemplar, but if you're interested, then these would be some other good associations to look at in further detail. So warfarin is an oral vitamin K antagonist, and the daily dose can vary 40-fold, and importantly, is the third most common culprit, leading to hospitalization for an adverse drug reaction. Warfarin as an anticoagulant thins the blood, and we measure that according to the INR, the International Normalized Ratio, and this biomarker is closely correlated with clinical endpoints. So a 10% of increased time outside the therapeutic INR range, which is typically between 2 and 3, is associated with increased risk of mortality, ischemic stroke, and other thrombotic events. And importantly, as the INR becomes supratherapeutic, i.e. higher, that increases the incidence of major hemorrhagic events. These data are from a UK prospective cohort study, and the Manhattan plot shows there are two very strong signals associated with mean weekly warfarin dose requirements. VCORC1 is the pharmacodynamic target of warfarin, and CYP2C9 is the phase 1 drug metabolizing enzyme responsible for the inactivation of the S-warfarin enantiomer, which is the most active form of warfarin. And actually, we can explain almost 60% of observed variation in mean weekly dose, with VCORC1 explaining 25%, CYP2C9 15%. There's another gene, CYP4F2, explains about 1 to 7%, and then there's a range of different clinical factors which are also associated, but to a lesser extent, interestingly, than these genomic signals. Warfarin pharmacogenomics has been extensively studied, and these here are the 3 most pivotal randomized controlled trials of warfarin to date. So the U-PACT study was carried out in Europe, and they demonstrated that a pharmacogenomic approach significantly increased the time in the therapeutic range, so that time that INR was in the therapeutic 2 to 3 range compared to normal clinical practice. Interestingly, at the same time, the COAG randomized trial was published, which was carried out in America, and they didn't find any significant difference in time in the therapeutic range between a pharmacogenomic strategy and a clinical algorithmic strategy. And the most recent and largest trial, the GIFT trial, again based in the US, actually was powered for a composite clinical endpoint, a 4-week major bleeding or a supratherapeutic INR, or venous thromboembolism or potentially death. And importantly, this showed that a pharmacogenomic strategy, in taking into account in all of these trials VCORC1 and CYP2C9, it significantly reduced the incidence of this clinical endpoint compared to standard practice. And therefore, on balance, it appears that warfarin pharmacogenomics increases the time in therapeutic range and decreases clinical events. And this slide from the U-PACT trial just demonstrates that the genotype-guided group experienced increased time in therapeutic range throughout the 12-week follow-up window, as well as avoiding the initial overshoot in INR, which the control group experienced during loading. And interestingly, when you look at which patients benefit the most, it's actually patients which have 2 or more of the variant alleles, either in CYP2C9 or VCORC1, who have an adjusted difference in time in therapeutic range of 11% compared to the control arm. So why did the COAG trial show no significant difference, whereas the U-PACT trial did show a difference? Well, this led, as you'd imagine, to an awful lot of discussion, and there are several potential reasons. One interesting reason is that the COAG trial recruited a more heterogeneous patient population in terms of ethnicity. So whilst U-PACT was almost exclusively Caucasian patients, COAG included 27% Black patients, 6% Hispanic. And actually, interestingly, the Black patients in the genotype arm actually did worse than the clinical arm. And this is likely because they were focusing on star 2 and star 3 alleles of CYP2C9, which are reduction of function alleles common in Caucasian patients, but actually in patients of African ancestry, there are other reduction of function alleles, so star 5, 6, and 8, and 11, which are collectively quite common, but these were not taken into account, and thus in this study, these patients were being erroneously conveyed as having a normal CYP2C9 extensive metabolizer phenotype, where in fact actually they had a reduced phenotype. It was just that some of these variants were not being considered in the study. So it's a lesson to us all that it's important to consider patient ethnicity when deciding which variants to test in pharmacogenomics. So just to finish off this Warfarin pharmacogenomics exemplar case study, whilst the previous slides have focused on randomized controlled trials, this slide just reports on a recently published real-world implementation study, which implemented the U-PACT pharmacogenomic algorithm, which takes into account the VCOR-C1 and CYP2C9 variants, in 3 anticoagulation clinics, compared that to 3 anticoagulation control clinics which use standard of care warfarin dosing, as well as other controls collected from routine electronic healthcare data. And this implementation study was carried out in the UK, and very similar to the U-PACT study, the final results demonstrate that the pharmacogenomic approach increased the time in the therapeutic INR range by 7% compared to standard of care. And thus, in summary, it appears that warfarin pharmacogenomics improves time in the therapeutic window and from the GIFT trial clinical events. And although not reported here, other studies have gone on to show that it's also very likely to be cost effective. So what are the barriers to implementing pharmacogenomics? Why are we not already doing it already? Well, in many ways, we have to just go back to think about those considerations I spoke about earlier. But a few particular barriers to focus on are the selection of a panel of clinically relevant genes to test, and also having appropriate IT technology in place and not disrupting existing workflows for physicians and pharmacists. Clearly, training and education is paramount. Cost effectiveness, cost consequence information is important, and real world experiences is very important as well, as previously mentioned when looking at warfarin. So to help try and overcome some of these barriers and catalyze the implementation of pharmacogenomics, the Ubiquitous Pharmacogenomics Consortium was founded and funded through an EU Horizon 2020 grant back in 2016, and it's a 5-year program running at the moment, which is implementing preemptive pharmacogenomic testing in real-world clinical setting across 7 different EU sites. The actual clinical study I'll cover in a minute, but it's called the PREPARE study, and we're evaluating both patient outcomes and cost effectiveness. So the Ubiquitous Pharmacogenomics Prospective Pharmacogenomic Implementation Study, the PREPARE study, has been running now for 3 years, and it underwent a block randomization process whereby each site was randomized to either start with standard of care or with genotype-guided dosing. And patients are recruited if they are starting if they're newly starting one out of a list of around 40 different drugs for which we have existing pharmacogenomic guidelines in place, and the patients in the genotype-guided arm, a blood sample is taken, it is genotyped locally, the report is produced and sent back to the patient's healthcare team, who then look at the report, and if there are recommendations, they decide whether or not they're going to implement those recommendations. In the standard of care arm, the blood sample is taken, but patients just receive standard of care, but that they will also undergo genotyping as well at the end of the study in order to facilitate the statistical analysis. And patients are followed up for 12 weeks, primarily to assess for the incidence of adverse drug reactions. And this slide just highlights the categories of drug for which patients can be recruited into the PREPARE study if they are newly starting. and also highlights the pharmacogenes for which they are genotyped for, and each patient is genotyped for all of these genes ideally, even though only one or maximally two of those genes will be relevant to the particular newly prescribed drug they are starting. But this panel approach is being used because if those patients then go on to be newly prescribed other drugs in the future, then they will already have this pharmacogenomic information in place. So the Ubiquitous Pharmacogenomics PREPARE study is implementing a genotype panel approach, but it's clear that technology is rapidly improving, and so this slide just highlights the number of nationally funded genomic medicines initiatives that are occurring, the majority of which are utilizing sequencing, whether that be whole exome or whole genome sequencing for their studies. And this plethora of sequencing data that's being made available is interesting because it makes it possible for the first time to start considering the potential impact of rare variants on pharmacogenomics as well. And that's clearly it's a new research area, but it's just something I'd like to touch on here. So just to touch on some work I've done myself within the research environment of the UK 100,000 Genomes project. This is just pilot data at the moment. It requires further analysis, but I've extracted variants, both single nucleotide variants and indels, from 195 selected pharmacogenes, both PK and PD genes, from 60,000 patients. Interestingly, of these variants, 94% of them were very rare, so present in less than 1 in 1,000 patients, and 97% of them were rare, so present in less than 1 in 100 patients, and thus only 2.8% were deemed common, so had a minor allele frequency of greater than or equal to 1%, interestingly enough. And when you then split it by SNVs and indels, I found that SNVs were slightly more likely to be rare and very rare compared to indels, but still the vast majority of variants were rare and very rare. When you then go on to look at the predicted function of these extracted variants, you find that the vast majority, about 97%, are classified as modifier variants, essentially they're intronic variants, and the remaining 3% can be stratified into either predicted to have a high, moderate, or low impact according to the sequence ontology hierarchy. When you then go on to focus on those 3%, you find that the majority are missense variants, so they're classified as having potentially a moderate impact, and almost a third are synonymous variants, so classified as having a potential low impact on the gene's protein function. And then you have a variety of others, for example, some of the high impact types of variants such as frame shifts and stop gain variants at lower proportions of this 3%. You can then go on to look at the relative proportions of high, moderate and low impact variants per gene, and so this is just a slide that shows that for select phase 1 drug metabolizing enzyme genes, and as you can see as a general pattern, the majority of them are moderate impact, reflecting that higher prevalence of the missense variants, followed by the low impact, which are predominantly the synonymous variants, and then the red bars showing the high impact variants, which are lower but fluctuate a little bit between these genes. And I should say these bars are not standardized as yet for the size of each gene. And similarly, the bar charts in this slide just depict the relative proportions of predicted high, moderate and low impact variants per gene, but this time focusing on some pharmacodynamic genes, either those involved in the cardiovascular or oncology systems. And interestingly, as you can see, particularly when you look at the oncology bar chart, that the red bars, those high impact variant bars, appear to be less prevalent, less conspicuous, and that's reflected in the table at the bottom, which looks at the mean number of high impact variants per gene according to category of gene. And for example, if you're looking for transporters, there's 24 on average high impact variants per gene, but for oncology it's down at 8.5, and actually neuropsychiatric genes it's down at 4.8, and presumably this reflects varying evolutionary tolerance for these high impact variants according to presumably the function of those genes and its importance for ourselves. So this slide just continues with my pilot data and again focuses on the phase 1 drug metabolizing enzyme genes, but this time is looking at the extent to which putative functional variation within each gene is attributable to either rare or common variants within the gene. Rare variants being those which had a minor allele frequency of less than 1%, and here are highlighted in red, and common being those with a minor allele frequency of greater than or equal to 1% and highlighted in grey. And in order to carry out this pilot analysis, I defined putatively functional variants as either those predicted to have a high impact on gene function or a moderate impact on gene function. And for the moderate impact ones that were missense variants, they were only included if the SIFT or polyphen algorithms predicted the variant to be at least possibly damaging. From my existing knowledge of pharmacogenomics, I did also carry out some manual curation to ensure that I included important other pharmacogenomic variants that we know about too. And what you can see from these graphs is that the extent to which rare variants contribute to this putative functional variation in the gene varies according to each gene. So for some genes, the majority of variation is attributable to common variation, but for others, it's almost exclusively or exclusively attributable to rare variants. Similarly here, this slide just highlights the putative functional variation attributable to rare versus common variants in the cardiovascular oncology selected pharmacodynamic genes. It is important to remember, though, for these analyses that the variants selected and incorporated were always exonic variants. And overall, when I looked across my 195 genes, I found that for greater than 50% of them, all of this putative functional variation is attributable to rare variants. And on average, the number of functional variants within this 195 gene set per participant was 37. And of these, 4 variants were likely to be rare, and therefore the proportion of genetic functional variation attributable to rare variants in these gene set of pharmacogenes is about 10–11%. Other estimates about the extent to which rare variation contributes to the function of pharmacogenes have differed, have been a little bit higher, 20–30%, but it appears to be around this sort of ballpark. And I'd still would contend that 10% is still not an insignificant amount of variation, and thus as sequencing becomes more common, hopefully we'll be able to interrogate these variants and link them to clinical outcomes. Second area with an eye to the future I'd just like to touch upon is the evolving area of drug-drug gene interactions. Clearly in clinical practice we're already accustomed to considering drug-drug interactions. However, if we implement pharmacogenomics on a wider scale, then there's going to be an increasing number of instances where a given so-called victim drug in a given patient may be potentially affected by both concomitant medications causing drug-drug interactions, as well as the patient's genetic makeup causing a concurrent drug-gene interaction, and collectively these can coalesce into drug-drug gene interactions, whether that be the perpetrator drug and the genomic variant acting on the same drug metabolizing enzyme or transporter, a so-called sort of one pathway effect, or actually if the drug is metabolized by 2 separate pathways, for example, but each pathway is affected, say one by the perpetrator drug and one by the genomic variant, then again you can have a substantial reduction in metabolism and a substantial potential increase in systemic exposure. And thus it's important for us to be considering these more complex interactions as we move forward with pharmacogenomics implementation. So just to highlight some recent analysis that I've undertaken and is as yet unpublished in a cohort of patients which were initially hospitalized with acute coronary syndrome, where we sought to identify pharmacokinetic interactions due to either concomitant medications or genomic variants. And if you look at the top pie charts, we identified 882 potential interactions in 503 of these 652 patients. And then when we stratified that into interactions which were just predicted to have a substantial effect on systemic exposure of the victim drugs, we could still identify 346 interactions in 252 patients. And interestingly, of these substantial interactions, the proportion of DDIs increases from 45 to 59%, but even more interesting, the proportion of drug-drug gene interactions doubles from 13 to 26%. When you then look at how many interactions could be identified per patient, interactions of any strength could go from 0 to 9, and of those substantial interactions, we still found patients with up to 5 substantial interactions. And lastly, when you just stratify by which enzymes or transporters are most likely to be involved, it's clear that CYP2C19 mediated interactions were the most common, and that's likely because these patients were predominantly taking clopidogrel, and also many were on proton pump inhibitors. But also, as you might expect from a cardiovascular cohort, SLCOB1B1 mediated interactions with simvastatin and atorvastatin, highlighted in the blue in these bottom pie charts, were also prevalent. Clearly, this is just identifying potential interactions. Future work will be required in order to assess the potential clinical impact of such interactions. So this just provides a summary of what we've covered today. I hope it has been useful. Thank you very much for listening to me, and have a good day.